Sometimes we do before time because the subject requires more time. And I also require 1 or 2 minutes more because I want to brief you what's happening in MMA next few days so that you don't miss them. Because some of our emails are going to spam, I believe, so that you have to pull out and decode it and it's not spam, then only it will come to your inbox. We also welcome all the viewers watching this program live, wherever you are from your own comfort. But we miss you. Start coming to MMA. I can assure you we'll have meet some wonderful friends and colleagues, a good networking place. But I can also assure you lovely refreshments what we give in MMA. Don't miss it. The next event is happening on the 17th of June. It's again very, very interesting event. India's IT revolution, the Maverick thing. We're also having a discussion on $1 trillion digital economy. We have got some outstanding speaker. Narayan Murthy, you all know him. Narayan Murthy is founder chairman of Infosys. He's going to be delivering the keynote address. Then we have Harsh Mehta and Mr. Ramani, who is a vice chancellor of Sai University. Lakshmi Narayan, former Cognizant, and Rajesh Nambia is the managing director, CMD of Cognizant in India. So we've got some outstanding people. We are going to discuss all about IT, how IT has got revolution, and NASSCOM, how it has come and changed the whole view, and how organizing such will help. So please, it's happening in person at MMA Management Center. We already have 200 plus nominations registered. Please do register in case you would like to come and attend this in person. Then on 23rd July, again in an interesting event, where from here the $5 trillion economy, because with the dream of our Honorable Prime Minister, where are we? Because the pandemic has really impacted, it's stagnated back for some time. But still, are we in the right track? And we have got some, again, outstanding panelists. We'll be discussing Mr. M.R. Shivaraman, he's a former Revenue Secretary, IAS retired, he will be there. And Arun Kumar, I know you must have seen his article regularly in Hindu, Dadiwala, you must have seen. Very good, very good analytical thinking. He writes so beautifully about the issues and policies. He's going to be talking to us. He is from JNU, New Delhi. We have Mr. K. Kumar. K. Kumar is a senior partner, leader of consumer industrial products and is head of Deloitte in terms of businesses. He'll be with us. And also Mr. Satyamurthy, who is also policy matters, will be joining us to lead the conversation. Then on Friday, 24th July, again we have an interesting event. We do an event on read and grow because we strongly believe if you're not a reader, you're not a leader. So we are trying to inculcate the habit of reading amongst our members. This program is very popular. A lot of people come and attend. We pick up some very interesting topic where the panelists come and discuss and how you can assimilate and keep it in yourself and how it helps you grow in whatever field you are. We have a topic of discussion. Start with why, how great it is, inspire everyone to take action. So very interesting. We have outstanding panelists. Babu Krishnamurthy, I think I'll walk a mile to listen to him. Very clear thought leader, investment consultant. Then Rajesh Balaji Ramachandran is a partner of Catalyst, and also Vijay Prabha Kamarakara is again founder CEO of Story Trails India Private Limited. These are the panelists. Please don't miss it. Then another interesting event I want you to really attend on 25th June. We are having a one-day conclave on reimagining businesses in New India. It's a paid program. Sometimes we also do paid program, and sometimes you must also attend paid program. You'll really like it because they are very good. I can tell you. We've got some outstanding sessions. Just to name a few, we have a very interesting person who is inaugurating and delivering a keynote address. Sri Sunil Mathur is the Director General of Income Tax Investigations, Tamil Nadu and Puducherry. Then we have a number of sessions, re-engineering conventional business using technology. We have again outstanding speakers. Then Make in India story for the next decade. Then we have a brand building for success. Vijay Parsadi, one of the outstanding thought leader in brand, is going to be talking to us. We have a session on tax technology, current and future trends. We also have another session on stock market opportunities in alternative investment. I don't know how many of you know what is alternative investment. Because with the interest rates coming down and alternative investment was the domain of only rich and wealthy. Now it can be brought down to the normal people like us also because they join as a group and group investment makes this investment very, very attractive. So we have experts who come and speak on this so that you have avenues. I'm not telling you must go to alternative investment, do that. But ultimately you have some knowledge about this in case you can take a very conscious and very decision based on scientific method so that you today the way the interest is coming down, the way inflation is going up, you need a constant income. I think this is some of the ways you can do that. Because I'm not telling you go and invest your every money in equity or Bitcoin or other thing and get into trouble. I'm not telling that. Understand our knowledge thing is to share the knowledge. You take the call what to do later. Then we also have an emerging technology-driven business growth. That's again. Then last but not least, we have a very interesting session on transition of family business to next generation, the integrity and nuisance of what's happening. Because family business get into trouble. Next generation it goes. And how we should start planning to pass the baton, how you should really professionalize your family business. So this is a very interesting event. It's a paid program. It costs almost 4,000. For MMA members, we already have 2,000 plus registration, 200 plus registration we have for this already booked. Because we are doing with another Augustine. We are doing this with another interesting organization in Chennai. I know you know of them. This is called Rajasthan Metro, ROA Metro. So it's all more business related people who are joining us together. We are charging MMA members only 1,000 rupees plus tax. Just the cost of the food and admin arrangements. And we're also giving some very, very attractive delicates kit. So do come, do network. We have some outstanding people. We also lined up program for the entire month of June. We are done, I just mentioned to you. We also program for July. Some important program which you must mark in your calendar. This 4th, we have a KCG Varghese endowment lecture, followed by dinner. Then 6th, we have again a conclave. On 6th, we have, you must come and attend that. Then 8th July, in fact, I want you to block your diary. 8th of July is MMA AGM. And we have some outstanding speaker. C.K. Ranganathan will be the chief guest. We have another keynote speaker who will be coming and talking on the subject. We have a MMA award function that day and followed by MMA AGM. Do come, and that's a time we showcase what we have done over the year and how we have been able to bring in a lot of changes and how we adapted to technology, and everything goes well. We also plan to launch a couple of new initiatives what we have done. Every AGM we do that. We do some new initiatives and which we do and showcase and launch it on important occasions. We have some surprise for you. Come and join us on AGM. We'll have some new technology initiatives what MA will be launching on that day. Then that's on 8th of July. Please do come. On 30th of July, Gangapriya Chakravarti will be talking. She's the head of Ford Chennai operations. And we have a number of events. Then Read and Grow is there. On 16th, we have capacity building for NGOs. So we have lined up program for the entire month. Again, I'll stop with July. I know you have come to listen to very, very important event today, and which is why you are here. I don't want to take your time. That's why I started 5 minutes before time. Still I gone. So still I got 4 minutes of mine. Let me tell you what's happening today. This is a topic when Air Marshal Matheswaran spoke to me. I can honestly tell you I didn't understand what he's talking about. Then I said, when I put my thought into that, this is a subject which is very, very vital today. Very, very important, because he sent me the theme note. After reading that, I understood, my God, how ignorant. I know this subject, but how ignorant we are that we are not able to understand what's happening in this particular subject. Potential of genome editing, actually. And many times we are troubling, what is it? How do you pronounce genome, your genome, or all other thing? But I think I pronounce correctly, genome. But it's a very important subject. We are indeed privileged, blessed to have some outstanding speakers we'll be talking. So potential of genome editing is food security, health, and surviving climate change. This is what the they'll be discussing. We have outstanding panelists, and I must tell you, you're all lucky to listen to some of the outstanding speakers. Especially, I must also tell you, our chief guest for this evening, who will be setting the contest and presenting the theme address is Dr. Chandrasegaran. Is a professor emeritus, John Hopkins University, Baltimore, USA. Thank you, sir, for joining us this evening. We have again equally a good and Air Marshal Matheswaran. I mean, he doesn't need introduction to MMA, dear friend of ours, and we do some. He's also founder and president of Peninsula Foundation, and we are doing a lot of events together. Air Marshal Matheswaran will be leading the conversation. Again, we have two young scientists who have done some phenomenal work in this field. He is Dr. Judy Gopal. He's a professor of research. Saveetha University, and Manikandan Muthu, Associate Professor of Research and Innovation from Saveetha University. Ladies and gentlemen, put your hand together to welcome our distinguished guest for this evening. May I request you please to take your place on the dais, sir. Come, sir. Yes, I think there's a big round of applause. They are going to talk some subject which we're all alien, we really do not know. I think we'll become more wisdom, more wiser by the time you step out of this hall to know about this subject. After listening to gentlemen, you will know how important this subject is. Air Marshal Matheswaran, as I mentioned to you, is not new to this audience, but it's always a privilege to introduce a colleague of mine, a veteran from Armed Forces, veteran from the Air Force. Air Marshal Matheswaran was an Indian Air Force veteran for more than 13 years of service, and he is the founder president of Peninsula Foundation, a policy research think tank based in Chennai. He's very passionate about, we already done a couple of events jointly with them, and he's a fighter pilot and an experimental test pilot and a fighter combat leader, and has flown over 40 types of aircraft. Somebody who would not know test pilot school there in Bangalore, AST we call them. Some of the finest pilots in the Air Force are selected to lead that, and he was a commanding AST, really to do a testing of all the aircraft to maneuverability and operations, and he was head of that. That requires a big round of applause because he's such a combat leader. Air Marshal also has a master's degree in military science, MPhil, PhD, and defense strategic studies from the University of Madras. And he has also done a senior fellowship from National and International Security from the Harvard Kennedy School, Governance and Harvard University. Air Marshal is a recipient of President Award, Adi Visit Seva Medal, then also Vaisena Medal VM, and also commendation couple of times from Chief of Staff. Very warm welcome to Air Marshal. Now the format of this program is Air Marshal will set the context, introduce the speakers. Thereafter, the keynote address will be delivered by Dr. Chandrasegaran, who is from Hopkins University. Thereafter, other two lady and gentlemen will also join us, share their insight. Thereafter, there will be a discussion. And if you have any questions, please do. At the end of the day, the mic will be passed on to you. Please share your questions so that, it'll be nice. Not your comments or lectures and other thing. If you want to talk to them something differently, the speakers are readily available for you for interaction with you the post event. We want you to relate your questions only to the topic what we are discussing, and really appreciate your understanding. And one more request, please put your telephones to silent mode. Normally I don't remind, our members are real gentlemen, they don't do that. But I thought once in a while to remind them, which is always good, so that by mistake you fail to pull off the thing. It really disturbs the flow of the speaker, that's the idea. Because the gentleman has come from USA, we must show how disciplined we are in India too. Over to you, Air Marshal Matheswaran.
Thank you, Vijay. It's always a pleasure to be here at MMA. And the Peninsula Foundation is ever thankful for the excellent support we get from MMA and KAS on various events that we've held so far. Very interesting events. And we will continue to do many more as well. It's a great pleasure to be here this evening. And I'm doubly happy because I'm hosting Professor S Chandrasegaran, who also happens to be my classmate, schoolmate, and Professor Emeritus at Johns Hopkins University, and to talk on his passion, very important subject on biotechnology, which is about genetic engineering and genome editing, which let me inform to the audience that he is the inventor of zinc finger nucleus genome editing. And that was done in the late 1990s. He headed a laboratory. While doing his PhD, he trained under Nobel laureate, Dr. Hamilton Smith, right? And he then headed a laboratory in Johns Hopkins University. I visited there quite often, and developed the genome editing technology. The ZFN is a costly process, but taking a lead from him, there are many other researchers who developed the subsequent versions which became more affordable technologies, which is Talon and CRISPR-Cas9 that you would have heard of, for which two ladies won the Nobel Prize last year. Logically, it should have gone to him. Well, so this is an extremely important subject. Along with him, we are privileged to have two young scientists, Dr. Judy Gopal and her husband and scientist, Dr. Manikandan, Muthu. Both have worked excellently well abroad. They spent more than a decade in Taiwan and in Korea. And they are biotechnologists with multidisciplinary approach. And therefore, they will bring in a rich, inputs through their experience and discussions for today's discussions. Their detailed biodata is available. Dr. Judy Gopal has worked in the Kalpakkam Atomic Energy department and on a material and biotechnology interface related doctoral program. And Dr. Manikandan has worked more closely towards the agriculture related biotechnology and of course interdisciplinary approach as well. Both have enormous research experience, and they are currently with Saveetha University, with whom we have an MOU signed just last month. Well, the subject, as I said, is extremely important because 21st century is going to be dominated by three important characteristics. One is the big data or IT or digital technology. Second, artificial intelligence and robotics. And third, and the most important one that's going to take center stage as we move along the 21st century is biotechnology. It's already doing that. And therefore, while Gupkar and Vijay Kumar said not many are aware of genome editing, it is important that people become aware of it. Right now in India, the knowledge is all about the controversies that have been raised with respect to the use of Monsanto's best cotton, BT cotton, and its linkage to the farmer suicides, etc., the seed monopoly and terminator technologies, etc. These have been in the news and most would have read about that. There is a difference between genetic modified organisms and genome editing. And that difference is what we are going to actually look at one. And what does that do? If you change fundamental DNA characteristics by inserting a foreign gene into the structure, does that become GMO? And if you are actually addressing an error or a problem in the existing gene through a synthetic genome editing, then that retains the existing DNA and continues to functionally evolve as naturally other organisms evolve over a much longer period. And what does this offer to the country? In the 1960s, India looked at ships from America that brought in PL 470 assistance, and poverty was endemic. And we addressed that through what's known as the Green Revolution. 50 years down the line, the Green Revolution has raised new questions. While our food production has grown enormously, we are now exporting grains. But at the same time, we are guilty of using excessive amounts of fertilizers, chemical fertilizers, which has led to a lot more problems. And therefore, today, many families in urban areas are looking to buy organic vegetables because there is a general prevalence of opinion and belief that the regular agricultural product is contaminated by chemicals. It has led to greater incidences of diseases like cancer, and therefore there's a lot of suspicion in people's mind. How do we address all this? Does genome editing offer a great solution? Addressing better yield, higher yield, retaining biodiversity, and not venturing into areas that could be seen as dangerous and creating more problems here. And this is the most important area that needs to be answered. The biggest question is genome editing safer, better, and more productive, particularly for food industries and farmers? And this is a question that needs to be, examined, and knowledge needs to be spread out. So without taking any more time, let me invite Dr. Chandrasegaran to deliver his lecture, and thereafter we'll move in with the panel discussion. Thank you.
I want to thank Mathesh and Jaykumar for inviting me, the TPF Foundation and MMA for organizing this talk. It's a pleasure to be here. Like Mathesh said, we go a long way back. Back in Sainik School, Amravati Nagar, we are classmates. We grew up together, okay? Especially wearing hard khakis and in a hard climate. I'm wearing khaki today, by the way, okay? I just want to talk to you a little bit about genome editing, revolution in life sciences, and I want to, just to make sure, disclosure, all the opinions discussed here are my own. It does not belong to Hopkins or anybody else, okay? So they are my personal views. I am fully retired from Hopkins. I retired in 2021. I retained an honorary appointment as Professor Emeritus there. So that's why I disclosure, okay? And I'm going to tell you a little bit about how we got started into this, what is genome editing, how we got started in early, how my lab got started into it, what we have done with regard to health. Then I'm going to ask my junior colleague, who is actually working in my brother's company, biotechnology company. Okay, they are doing some interesting research, so he's going to give about 10 minutes talk. Okay, so that's how I planned the talk. Okay, what is genome editing? It's also known as gene editing, because let's say there is a mistake in a gene and you get a disease. What you want to do is somewhere go, correct that mistake, hopefully it will produce normal protein and you'll be fine. Okay, so we want to edit that mistake. Okay, that's what we want to do. Okay, it's called genome editing because we are doing it at the genome level in a cell. Okay, some people refer to as gene editing because we are targeting a particular gene to correct. Okay, so it's also known as gene editing. Okay. So whenever you take a bacteria culture, you add some donor DNA, it does two things. One, it can randomly integrate that piece anywhere within the genome, or it can go to a site where it has a sequence similarity to the donor DNA and replace the genomic DNA with the donor. That's called homologous recombination. Okay? In bacteria and yeast, the recombination, homologous recombination occurs at a very high rate. Okay? So they are very useful from that regard. But once you come to mammalian cells, especially human cells, the homologous recombination takes place at only 1 per million treated cells. So it's a very rare event. So it's very hard to isolate those cells. Okay? In mice cells, which are slightly better, you can do it by positive and negative selection, isolate those corrected cells. Okay, that was what was done by Mario Capecchi, for which he won the Nobel Prize, okay, a while back. Okay, from that corrected, he did that using embryonic, mouse embryonic stem cells, so you can regenerate mice from those cells. So you can form transgenic animals. That's why that research became very important. What we wanted to do is correct a mutation within a human cell. Okay? So biologists have known for a while, if you want to stimulate homologous recombination, all you have to do is make a break at that locus. Okay? At that site, if you make a double-stranded break, you can stimulate recombination. So, when you have a double-stranded break, the homologous recombination increases 1,000 to 10,000 fold, even 100,000 fold sometimes. So essentially the goal is to stimulate homologous recombination. So how do you do that? When you're talking about DNA, how do you break DNA? You're looking at molecular scissors. At that time we entered the research, there were no scissors available to make a precise cut within a genome. Remember, this human genome is 3 billion base pairs. To make a precise cut, there are about 123,000 genes within the genome, and to make a precise cut at one place, you need high specificity. The restriction enzymes are the molecular scissors that bacteria produce, but they produce it to protect themselves. So these sequences occur more frequently. You can't use them for making gene targeting. So we said we had to come up with a way. So that's the problem we face, and we decided to approach that. So what happens when you have a double-stranded break? Okay, so the repair cells don't like the double-stranded break because all the information, your genome encodes the whole program of the life cycle. So what you want to do is immediately patch up that break. So it tries to do it by two ways. One is through non-homologous enjoining. What it does is just patches it up. Okay, that process is error prone. So it makes mutation. So you end up with a pool of mutations. Okay. In the other case, you give the Ziffens, Talens, or CRISPR, you make the double-stranded break. At the same time, you give the donor. As long as your donor DNA has homology on either side of the break, it will integrate whatever donor you have into the genome and correct. Okay? But you can also introduce whole genes as long as they have homology. I need to know pointer, right? No pointer? Okay. Yeah. As long there is homology on either side, it can integrate the whole gene. When you do that, this is, you can introduce novel function, or you can introduce novel traits. This becomes GMO. If you are just disrupting the gene, you are knocking out the function. It's a loss of function. That's non-GMO. At least that's how FDA characterizes. So when we started the work, we had to make a molecular scissors that cut okay, I'm used to. Let me do it here. I can't go back? Okay. So when we came to the research, what we did was we looked at this enzyme. This is called a FOC1. So we said this enzyme has two parts. One for recognizing the GG-ATG sequence. So it has a DNA binding domain. The other one has a cleavage domain. That does not recognize any sequence at the cleavage site. Okay? So the idea was you have two parts. It's linked together by a linker. You can separate the two. Then you can replace the recognition part with a designer enzyme. So we want that new DNA binding protein should be something where we can control the specificity. So at that time, we chose this protein, zinc finger protein. It's an antiparallel beta sheet with the alpha helix, which is held together by a zinc atom. And this zinc atom coordinates its two cis amino acid residues and two his residues. Okay? And it's in a compact form. And the business end of the molecule is actually at the amino terminus, okay, those make the base pair contacts. Each of these motifs recognize 3 bases, okay, 3 to 4 bases. There are only 64 possible triplets in DNA. So if you have 64 of these modules, we thought you can, like beads, you can collate them, make them recognize a big site, then you can fuse it to the nucleus domain and make a new protein. That will be, by making a long recognition site, you can make it cut very specifically at within a genome. So this is what the next slide shows you, how the recognition takes place. Usually they recognize the primary strand, the amino acid contact the primary strand. If you have aspartic acid at 2 position, they make a cross strand. So if you don't have it, we thought without aspartic acid at 2, you should be able to have 64 possible zinc finger motifs. So you can mix and match, make them recognize long side, then take that, fuse it with the FOC1 nucleus domain and make a new enzyme. It turns out you can do that. Here you have 3 of the modules linked together and they recognize a 9 base pair site. 3 zinc fingers recognize 9 base pair. Since they all have aspartic acid, there is 1 more contact, so actually recognizes 10 base pairs. Okay. Did it work? Yes, otherwise I won't be here talking about this. So we made 2 of these. Two 3 zinc finger proteins. Actually, it's shown on the right hand side. You can see lane 3, where you are purifying the protein and cutting the lambda DNA and running it on agarose gel. You can see 2 smaller bands. Then on lane 4, you see 2 different bands. Again, 2 smaller bands. So we use 2 different specificities, so these enzymes work. Actually, we are even luckier than that. What happened was, of course, if you want it to be very specific, to make a specific cut within a genome, you need about 18 base pair recognition. Okay? So that, because just doing the probability 4 raised to the power of 18, it becomes very rare within the genome. It turns out that FOC1 nucleus domain has to dimerize to cleave. It has to dimerize to cleave. That meant its inverted repeat is the best substrate for this enzyme. So that immediately makes it 18 base pair. So it's a rare, so we can use this to make rare cuts within a genome. Of course, the other advantage is you can also cut a heterologous site by giving 2 different zinc. That's shown on C. Okay. Now, fine, we showed that this all works well within test tube. Then we had to test it. Does it really do genome editing? So we tested it using a model system in frog oocytes, frog eggs. So what we did was we made a plasmid which contained 2 TET genes in the same orientation. It's a small molecule, so you can easily isolate from the cells. It has a unique site, so you can linearize it and analyze it in gel. So you take that plasmid, inject it into the frog oocyte, wait for 4 to 6 hours. It will form chromatin structure. Then you inject your zinc finger nucleus, which we created. So it can do only 2 things. One, it can cut, or it doesn't cut. If it doesn't cut, when you isolate and linearize, you should get a 5.6 kb fragment on a agarose gel. And if it cuts, if nothing else happens, you should get 2 fragments, 3.6 and 2 kb fragment. But there is one other possibility, because you have TET genes in the same orientation, they can undergo recombination, homologous recombination, because sequence similarity between 2 TET genes are 100%. So they can recombine and give you 1 copy. When you linearize, you'll get a smaller band. And that's what this next slide shows. You can see the uncut is the substrate when nothing happens. When recombines, you can see that you get this smaller fragment. Okay, so it shows two different enzymes. One has QQ inverted repeat. When you add that Q enzyme, you get the smaller recombined product, REC, as shown. Then you have the KK substrate, then you add a K enzyme, again that recombines CK. Then you have a heterologous site, then you add only Q, it doesn't undergo recombination. Then you have again heterologous, you add the other enzyme, it doesn't undergo. But when you add both enzymes, it undergoes recombination. So this was the first experiment which says you can use ZIFFens to do genome editing. Okay. When we did more and more work, one of the problems came up. The problem was ZIFFens we also saw were very toxic. The reason for them to be toxic was many of the modules reported in literature don't exactly recognize 9 base pairs. Many times they recognize only 2 bases as shown in ZIF1 and ZIF3 in the next slide, right? The center base is not recognized, so they can be any one of the 4. So that leads to off-target effects. So one of the things you really have to do is you have to choose your ZIF modules with high specificity. If you don't do it, you'll run into trouble. Okay, there are a lot of ways to avoid this. One is just increase the length, increase the number of modules. It will recognize a bigger sequence. It will be more specific. Then other way is also if you want to avoid cutting it inverted repeats, which can happen within the genome for the enzymes. So you can do that by creating a heterodimer site, okay, at the FOC1 interface. That's been done and that's been published. Now I want to show you here. Here, these are the applications that came out of ZIFFens, genome editing. Okay, this was till 2012. All these things which were not possible before became possible after the development of ZIFFens. 2012, guess what? CRISPR came in. So actually ZIFFens laid the foundation for genome editing. CRISPR came and made it easier. So after ZIFFens, we did it in 1996, Talens came in 19. Talens is very similar to zinc fingers because Talon is a different DNA binding module. They are also similar size to zinc fingers, but they recognize only one base. So all you need is 4 different modules. So you can mix and match. Then again, they've also fused it to FOC1. It forms the same strategy as FOC1. Okay? But unfortunate for Talens, 2 years later, CRISPR came. So in this case of ZIFFens and Talens, you have to do protein engineering, which is very difficult to do. Okay? So that's why it's very costly. So for making one, in the early days to target one gene, it cost about $10,000. When Talon came in, it dropped to $5,000. When CRISPR came, it dropped to $50. So that's the difference. Okay? In the case of CRISPR, the protein is always, the nuclease is always the constant. The target recognition is done by RNA. Right? The target site is pulled apart and the RNA makes the recognition where it's shown by N on the top. I'm always having problems. Anyhow, by the RNA, it's called guide RNA. That does the target recognition. Okay? But as guide RNA is very easy to make. You can order them. Okay? You can order the DNA, put it in a plasmid, which then you can make the RNA very easily. Then there was an explosion. Everybody wanted to use this. That's how, for that work, Doudna and Charpentier were awarded the Nobel Prize for harnessing Cas9 to do genome editing. Okay. Now, these are some examples of how genome editing has been used. Here is one on the top. A panel shows edited white button mushrooms. Okay. I don't know if you guys if you have all peeled potato and leave it out, after some time it will become very dark, black. After a while you don't feel like eating them. Okay. So what they did here was, to avoid that, they knocked out polyphenol oxidase. It's an enzyme within there. Okay. And there are 6 isoforms. They knocked out one of them. So now the mushroom doesn't darken. Okay. So it's very sumptuous. You want to cook and eat it. The second one was done by the flower. The flower on the right hand side, violet, is produced by anthocyanin gene. Okay. So it produces the pigment. That gene produces the pigment. This is Japanese morning glory flower. So the Japanese scientists targeted the anthocyanin gene and knocked it out. Then from that you can see it becomes a white flower. Okay. So it doesn't produce the pigment anymore. So it becomes a white flower. Okay. Then you have the cow here. Okay. So beef industry is a big thing in US. Okay. A lot of people eat beef, and when you have cows have horns, they hurt workers. That can become a big lawsuit, and they can also hurt other cows. So you don't want them. What they do in the old days is pull out the horns. Okay. Which is very bloody and messy. Now you do it in embryo. Okay. They knock out the pole locus. Okay. It's a locus, a chromosomal locus, which gives rise to this horn. So if you make a small deletion there, it doesn't produce the horns anymore. Otherwise the cows are sad looking, but they are all very healthy. Right? And on the other, the last slide, D, I saw there are 2 snails. So if you take the snail on the right hand side, it's a right-handed screw. If you look at the shell, it's a right-handed screw. That's because it has a gene called LSDAA1. So some scientists knocked out that gene. Okay, when they knock it out, you can see its progeny on the left. It has a left-handed spiral. The shell is a left-handed spiral. It's a left-handed screw. Okay? So you can, by targeting one gene during development, you can change the whole body plan. Okay? And you can see why is this so important. Actually, in humans, you can have organs. There are some instances where organs are rearranged. Right? So these can help in those studies. Now, my lab, after doing that, I am at the medical institutions, so I used to be, retired now. So I surrounded colleagues who are all interested in medicine. So my thinking was all, how do we use this technology for correcting disease correction? So we thought we'll go through this way. The idea is if there is a mutation which causes a disease, can we correct it? Right? So that's what we wanted to do. So we chose, we want to do it in stem cells, so you can put it back, take a patient, take his own cells, then correct it and put it back. So it's autologous transplantation. So you don't have any immune rejection or any other problem such as that. So what we wanted to do was, we took 3 diseases, sickle cell disease, which has a mutation, where in the beta globin gene, there is a mutation, GAG is converted into GTG, which changes the amino acid, glutamic acid to valine. Okay? That affects the hemoglobin. That makes the cell sickle. That's your red blood cells sickle, they clog together and can block arteries. So that's a serious disease. It's very common in African, but also in India, it's pretty common, I think. Then we address the second disease, which is the Gaucher's disease. It's a lysosomal storage disease which affects white blood cells. So it affects their mobility. Okay. So if the enzyme is mutated, they can't move. So you can't fight all infections and other things. So it can be really bad. And of course, third one is the most difficult one is cystic fibrosis. But our lab has only addressed them in the lab. We have not. Can we do them? So the approach we took was very simple. You take the cell, but usually somatic cell, which is differentiated cell, you can edit them, but they won't survive too long. So you have to keep on doing them. So what we thought is take the somatic cell, reprogram into a induced pluripotent stem cell. This is using Yamanaka's work, for which he won the Nobel Prize. And then you do gene editing. Then you differentiate it into the right human stem progenitor cells and infuse it back into the patient. That's what we wanted to do. Can we convert them into, can you take the differentiated cell, like take your fibroblasts from your skin, convert them into human induced pluripotent cells? We could do it. You can see cystic fibrosis is converted into CF-HIPase. See, the top panel shows differentiated cells. When they become induced pluripotent, they become like a bag of cells. And you can see all the four. We put all 4 stem cell factors into those differentiated cells and reprogram into pluripotent stem cells. Okay? And we show after reprogramming, we remove all those factors. Then once reprogrammed, they stay reprogrammed. Okay? Now, then we do the editing. So we did that. So it shows you on this panel on your left, below the gel graph, you can see SCD-IPSC. You see only GTG. Only T is there, that red line. Okay? That's a sequence trace. So you can, that sequence is only T. When you see on the right hand side, you see CRE2E, you can see 2 colors, red and green, where you have T in it. So each cell has 2 copies, one came from the father, the other from the mother. So you have corrected one of them, one allele. Okay? So they are corrected. And the bottom panel shows they remain. Then we differentiate. We show that it produced erythrocytes. Then we look at how well look for HBG, globin expression, by RT-PCR. You can see in the sickle cell you don't express, almost nothing is expressed. When you correct one allele, you get 100-fold increase in the expression of beta globin. Of course, if you correct both copies, you'll get complete expression. So Gaucher's disease, this is also, I told you, the second one we worked on. There are 3 types of manifestations of disease. Type 1 is very low, it's not very serious. Type 2 is the most serious form. Type 3 and type 2 give neurological function. This is what the enzyme does. It actually breaks down glucosylceramide and glucosylsphingosine into ceramide and glucose and pyngosine. Then they are recycled back. If you don't do that, if the enzyme is defective, then your lysosomes don't function. So your cells cannot move. So if you guys, white blood can't move, then you can't fight infection. And it damages your liver. Okay? So the same strategy, we take GD fibroblasts, we make induced pluripotent cells, we correct them, then we differentiate in macrophages and look at them. Again, coming to the right panel here, you can see DG. GDHIPAC, CCG. This is a sequence trace. When we correct one allele, one copy is corrected. You see both of them, T and C. Then we also have both corrected, biallele corrected. You have, right? Just both corrected. What does it do to the actual transcripts? You can see on the left panel here, RT-qPCR GCase transcript, macrophages. We are looking at there is hardly any transcript. When you have one allele corrected, you produce almost 10-fold more. Then you have maximum production when both are corrected. Then you can see the protein also. Western actual enzyme production, you can see when you have corrected, you have produced a lot of it. So what are the challenges with genome editing? Genome editing, the big problem is off-target cleavage. Okay? So they can cut somewhere else. Although you can correct, you don't want it to do. It's like a knife. When surgeon is, doing his operation, if he cuts it somewhere else, you're in trouble, right? Same thing true here. So when you do genome surgery, you want it to be very precise. But there is off-target cleavage in all the three tools. Okay? And also efficiency of cleavage is low. Still very low. So we need further improvement. We have to develop more safe and efficient delivery. Not all cells work well. Some cells work better than others. Okay? Different methods, talents give it different results, Ziffens give different result, CRISPR gives different result. Of course, the last one is most important. Right now, this is a low estimate. Per treatment, it's about $1 million. I don't think many of us can afford it, right? So the high estimate is $2 million per treatment. So, of course, in the autosomal recessive disorders, recessive diseases, you have to have both copies of the gene. For a person to have a disease, both copies have to be mutated. Okay? So all you need to do is correct only one copy, then the person will be fine. In the case of dominant disorders, both copies, all you have to do is have one copy mutated, you'll have the disease. So when some person has biallele mutation, you have to correct both of them. That biallele correction is even more difficult. Okay? The biggest controversy about genome editing is this one. Human embryos. Should we to edit or not to edit human embryos? In USA, genome editing of human embryos is strictly prohibited. Okay? No, not allowed. You can't even create embryos to do the research using federal funds. Okay? Then, of course, you'll have all the other. My personal view, it will eventually happen. Maybe not this decade, may not next decade, maybe 50 years down the line, it will happen sometime. Okay? The other problem you have is people wanting to do cosmetic changes. That is not good, because whenever you do changes in embryo, you are talking about future generation. Right? It will be passed on to your progeny. You don't want that. In the therapies I talked about, you are taking that patient, you are doing everything to that patient, you are affecting only one person. So that's not a big deal. But if you are going to affect all future generation, you have to think carefully. So, but that has to, not just to do cosmetic changes. If like I said, US National Academy of Sciences has made it clear it should be done in only more serious cases, only when an alternative is not available. Okay, but right now none is being done. Right now we don't have to worry. I'm sure some of you have seen these movies, Gata and Boys from Brazil. Okay, genetically modified organisms which are knocking out genes. They do not require US FDA approval, okay, at least so far. So even that polled locust, these cows without horns, they have, you have seen cows with mini horns, they arose about 500 to 1000 years back. They are still being propagated, but still they are pending, I think, in US. Organism, the moment you put a novel function, introduce a novel trait, you have to get US FDA approval. I'm not too familiar about Indian thing. I think that's where I'll stop. If it were up to me, I will set up a training center to train these two technologies. My lab has also worked on synthetic biology, where we have synthesized whole chromosome. Some of you might have heard about SC 2.0, synthetic yeast 2.0. So these two technologies are going to dominate next 50, 100 years. So if it were up to me, I had a lot of funding, I would train every high school student, take them and expose them to these things. Okay? And it's the future. Okay? You have to have these. And also set up a good training center for genome editing, focused on only genome editing and synthetic biology and bioinformatics. Okay? I let my colleague present about 10 minutes what he has done at PBL in my brother's company relating to agriculture.
Thank you.
I can just sit there. No? Sorry, long.
Yeah, Hari, please go ahead.
Good evening to all of you, and thank you, Professor Chandra, for this opportunity. I think from Professor Chandra's talk, you might have already known the basics of genome editing. I think from Professor Chandra's talk, you might have already known the basics of genome editing, and he has already discussed about the potential applications of genome editing or gene editing in the field of medicine. But here I'll be discussing about the work we were doing at Pondicherry Biotech Private Limited, which was located in Pondicherry. And here we are mostly working on the genome editing applications in plant systems. So the topic I'll be discussing here is in vivo genome editing of Indian tomato plants. And the work was carried out by me and my junior colleague, Mr. Prakash, under the mentorship of Professor Chandra. So this is the basics of genome editing, how the genome editing uses the DNA repair pathways. As I think we have already discussed about this. When there is a double-stranded break in the cell's natural repair machinery the cell's natural repair machinery takes over the repair mechanism, and there are like two repair mechanisms. One is the NHEJ based method, that is non-homologous end joining, and the other method is the homology directed repair. So if the natural repair, mostly more than 95% the NHEJ method only occurs, whereas HDR occurs very less frequently. So when there is NHEJ method, it tries to mix match. So it either inserts some bases or deletes some bases, and then it ligates the DNA. So you will get a knockout function. So the knockout means the protein function will be knocked out. So mostly these are called as non-GMOs because we are not introducing any foreign gene or any gain of function mutation. Whereas the HDR method requires a template which can be used for DNA repair. So either it's like a gain of function mutation. So you are introducing a new function. So this technique is called as GMO, that is genetically modified technique. And as the title mentions, like we are working on the tomato plants. If you look at this picture, it's a domestic wild plant. It's called Solanum pimpiliformis. This is a very wild type. It was like 1,000 years before. And in the domestication process, the size of the tomato, if you look at this A to B, it gets increased. And this is the latest variety we are getting. So if you look at the increase in size, so in the reverse genetic approach, the scientists have used the reverse genetic approach to find out which are the genes responsible for this increase in size. So if you look at the increase in size, so improving yield by increasing the size of produce is an important selection criterion during the domestication of fruit and vegetable crops. And it can be increased by increasing the number of fruits also. These are the two approaches. So by reverse genetic approach, the scientists have identified some genes which are involved in the fruit development. So the genes are like LC, it's responsible for locule number, and FAS is for fasciated genes, and ENO is the excessive number of floral organs. And these are the genes which are involved in fruit development. So these are the naturally occurring FAS mutations which are observed in the tomato plants. So what we have done at PVPL is, so we have identified a target gene, and using a genome editing technique, so we have targeted a gene and then we looked for the phenotypes, fasciated phenotypes. So what is happening to the fruits? So if you look at this, this is the control plant where we have not done anything. So it's a control, and this is, we have identified like a gene, and then we have designed two target regions. So this is the target region 1 and this is the target region 2, and then we have combined both the targets in a single construct. So if you look at the fruits, these are taken from the field only. So these are control fruits are like normal, whereas if you look at the target sites, the fruits got fasciated, fused. So this is after harvesting. So if you look at the morphology, so compared to the control plant, here you have got lots of fasciated fruits, that's fused fruits, due to the disruption of the target gene. So if you look at, so when there is an increase means it is due to the increase in the locule number, as we discussed in the first slide. So we just look for the locule number, increase in locule number by using the target. So these are the control plants where you could able to see only there are like 3 to 5 locules in the control plant, whereas in the mutant plant, the locule number got increased. Here it is 9, 10 and 11. And in our experiment, this is the maximum number of locules we have got. I think it's around 17 locules here. And I have to emphasize that these results are like very preliminary, and we have to do like advanced research to confirm this, and we have to proceed this to the next generations to check whether these characters are inherited to the further progenitors. And then, so like we discussed about locule number, there is an increase in fruit count is also like a increase in fruit yield. So these are like, this is the control plant where we could able to see only few fruits, and this is the target site 1 and target site 2, and then this is the compared. Whereas compared to control, here in target site 1 and 2, the fruit count got increased. So here it's in the chart. So for example, if you look at the mean fruit number count, this is the control plant, and this is target site 1, and this is target site 2, and then this is the combined one. So you could able to see that the fruit number almost got tripled here. Actually, when I said in the title, it's Indian tomato plants, we have selected 3 varieties of Indian plants. One is like a Pusa Ruby from the Indian Agricultural Research Institute, IARI, and then second one variety is a PKM1 from the Tamil Nadu Agricultural University, and then one variety cherry tomato we got from online. So this fruit count is for the Pusa Ruby variety. And this is for the cherry variety, and this is for the PKM. So we could able to reproduce the results where you could able to see in the target site 2, the fruit number almost got doubled. And we have already mentioned that these are like very preliminary results, and then we have to prove that these results get inheritable in the subsequent generations like M1 and M2. Since if you look at the life cycle, it takes almost 6 months. So we started this, now we have the fruits. So like we are like proceeding to the further generations. And then we need to establish the genetic basis of the results. So we have to confirm through the molecular analysis, like sequencing and other things. And we are also exploring the use of local algae for biofuel production, and also a duckweed variety for biopharmaceuticals production. And we are also interested in developing herbicide resistant and salt tolerant rice. Like we are working on that using the genome editing approach. And this is our team working at Pondicherry Biotech, our Professor Chandrasegaran mentor. And this is our President and CEO, Mr. Chandrasegaran, and Junior Scientist, Mr. Prakash, our Director, and then our Lab Assistant. Thank you.
Thank you, Hari. Come, Dr. Muthu. And Judy, come. Yeah. Okay, it's been an excellent long coverage, and One of the important things about the genome editing and genetically modified organisms is that the laws and the regulations across the world, for the very reasons that Dr. Chandrasegaran explained, the challenges with respect to human use in animals and human beings and for medical purposes, have a lot of issues related to control, ethics, as well as challenges, medical challenges. And therefore, in most countries, as you pointed out about US, this is still a no-no. In India, it's completely not allowed. But for plants and agriculture, this is coming in a big way. And I think, as Hari pointed out, as tests and experiments go along, it needs time. And therefore, more data has to be generated to be able to become commercially viable for farmers as well as industrialists. So, in terms of regulations, the government of India brought out, it has very stringent regulations with respect to genetically modified organisms since 1980s, very, very stringent. And the only GMO crop that was allowed is Monsanto's best cotton seeds, which is ultimately over 20 years, it's raised a lot of controversy right now. And therefore, they're quite, cautious. But in March, they introduced new regulations in the context of new technology of genetic editing, genome editing developments. And therefore, the government has given very relaxed regulations with respect to what's known as SDN1 and SDN2. These are ones where external DNAs are not introduced, external genes are not introduced into the thing. It's only gene editing as explained by both Dr. Chandrasegaran and Hari. And genomes, and instead it would rely on reports, instead of going through a bureaucratic and red tape procedure of, a long process of clearances. These are being very relaxed rulings, which will allow the gene editing to be adopted immediately into the agricultural industry. So this will help the breeders and researchers in a big way. Similarly, the US has relaxed it significantly. In 2019, 7 crops were cleared, gene-edited crops. In 2020, it's risen to 70. But European clearances are merged. They still deal and consider gene-edited organisms as equivalent to genetically modified organisms. And therefore, it is quite strict. So in this context, and with what Dr. Chandrasegaran has covered, we'll invite comments from Dr. Muthu, who deals with the agriculture significantly. He has his own test farm in Manikandan. He holds 3 patents in Taiwan to his name. And let's invite his comments, followed by Dr. Judy after that.
So, good evening.
About 6 minutes each, that's right.
Okay. So, good evening to you all, and I'm really happy to be part of this ceremony here, to interact with Professor Chandra, and also I'm happy to be with our uncle. And also Dr. Judy and Captain Vijay Kumar. So very happy, and actually it is quite really fascinating me, when we were in masters, we studied the textbook of nucleases and all those things, and the real people behind it, to bring all those into limelight is here. So I'm interacting with the eminent scientists. So it's really happy, and I'm coming to the topic of gene editing. As both of them described about the gene editing technology, bring it into field level is having a little difficulty. We do not know how many generations, since it is mostly concentrated on single allele process, and we do not know how far it will resist to present in the organisms, and how many generations is passed through the same traits. So that difficulty we face, and also this what I understand from their talk is like this can be easily hijacked by some other people to create superbugs and that kind of things to spread it to the society. So that there could be a possibility for that too. So these are the things which came into my mind. And the other thing is like the regulatory bodies. Is it really as simple as we talk here? Like, can we get the permission from the regulatory bodies? Because I have come across, the government bodies. We have to undergo six bodies permission to really bring about genetically modified or genetically modified organisms. If we bring them with a good trait or so on. So we have in India 6 different bodies like Recombinant DNA Advisory Committee, Institutional Biosafety Committee, Review Committee on Genetic Manipulation, Genetic Engineering Appraisal Committee, State Biotechnology Coordination Committee, district level committees. So we have to pass through all this in order to get permission for a single, our achievement, in fact. So these are all the difficulties we face here. In fact, what I did was, when I came from Korea, the peanut sizes are twice the size of our Indian peanuts. So I am totally admired with those peanuts, and I brought some seeds, like it is climate oriented, like those grow in a cold climate. And then when we bring that to our condition, it is very difficult. Like I bought 1 kg seeds, and out of it, like I got only 2 or 3 plants grown. So from that, I just grown those seeds, plants, along with the plants of our Indian varieties, indigenous varieties. So what happened was, like the next generation, I got so many plants with a very high yield. The character is still retaining, but I'm literally confused, and I'm scared to get permission from all these bodies. So the process is still going on.
Are they genetically modified? Or
It's not genetically. I suppose maybe some crossbreeds, naturally, natural crossbreeds. I have to verify the genetic makeup, the genome of the new peanut variety, which would be very useful for farmers, because one peanut plant may produce more than 130 pots of peanuts. It is amazing, excellent result. But I'm literally confused to get the permission from this. There are a lot of rules and regulations, laws we have to oblige and to bring it to the farmers. So these are the things I come across, and also we need to see on the biodiversity perspective, like a system as a whole, the diversity as a whole can sustain itself. So we were told like the Prosophis juliflora plant. People may know that it's a plant originated from Cuba. Still, Cubans are using it for generating energy, like power and all those things. But that was introduced to India, but it became invasive species now. So we cannot eradicate those plants from India. So what I suggest is the biodiversity itself, organizes of its own. But when what we have to do is, we have to thoroughly study before introducing any kind of GMOs or any genetically modified organism through gene editing or anything, thoroughly studied in the perspective of biodiversity. And that is more important in my view. by considering all the species around it. So if it become invasive, then it would be dangerous to the ecosystem, and it can even alter the climate also. So it should be taken care of. That's what my opinion here.
Thank you. Thank you. Judy, your comments?
Hi. And so actually I thought it's more about, in my case, I thought I'd just comment on the theme which is running here because I'm really not a specialist on this, and I thought that it's a general audience mostly, so I thought I'll just comment on the theme. So the first thing I also thought was Marshal Matheswaran, he said, and also like they have all key interests, also Captain Vijay Kumar, they all said that this is a term when I heard the term, I was like shocked. Starting as a biologist, for me, when I heard this term, I was shocked. Because this has started a bit later down the line, far from when we did actual biology, and we've kind of like outgrown everything. And then, so when we heard gene editing, it was like really new. Editing, yes, editorial, yes, my father works with the editorial in the Hindu. So yeah, we know what is editorial, we do a lot of stuff with the text. But with genes, that is something that you do things with the text, you just delete something, you just take away something, it's not gonna hurt. But you do something with the genes, you're going to hurt. And it's going to hurt not just this generation, it's just going to perpetuate into the progenies and into the next generation. So this is something that is relatively more serious, and something that we all need to have a consciousness and an awareness, and not just say like, oh, that's the job of a biologist. So when when especially Uncle introduced the theme, I just thought, I was really humble. Because I thought like, here is something which is so important, and there's so much work going on, and then you say that this is not my field, I changed domains, I'm working on something else, and what is close to humanity is close to us, and it should always be close to us. Be it what level of scientist you become, or to which discipline you switch, we need to stick onto things that are close to humanity, and this is, yes, something very close to humanity. And I'm so glad that Madras Management Association has thought of doing something, talking about something. Maybe obviously these are like people who've been in the aviation industry, and I really do not know why they would talk about this, but then yes, they are talking about it because it's very important to the society. And so I think this is something that all of us, we can just Google it. We could just have a broad knowledge about what's really going on here. So as the theme goes, it says like food security, potential of genome editing, and in food security, health, and surviving climate change. So I just did a small search. And I just thought that the terms were a little bit not related, like genome editing. Oh yeah, we do genome editing, and that is like you would have a disease-free environment. You would have people who are struggling with disease, with genetic disease. You would be able to impact them, and then you could change a gene, and then in that way you would be able to get them out of the disease. Yes, that's very welcoming. And then you go forward, you look at agriculture. Yes, you do a bit of genome editing, and then you do a little bit of changes in the genome, and like the researcher who showed just now, you would have things produce products that would be much better than what you're using. Yes, these are exciting. But when you do these changes, what are you changing? And are you in a position to change it? Do you have an authority? We heard a person with authority, and he started doing changes, and that has positively impacted the society. So I saw a beautiful article, which really challenged me. It said like genome editing, hi-fi, everybody's talking about it. Is it a boon or is it a bane? It's food for thought. Is it a boon? We're talking about, wow, we can do this, I can do this, I am Superman, I am Superwoman, I can do this. Is it a boon? Will it be a boon? Maybe it can get me a few laurels, but then will will it be something that would impact negatively the future generation is something that is a question that we need to question ourselves whenever we play with genes. We're not playing with toys, we're not just playing with our lives, we're not just playing with whatever is around us, we are playing with stuff that could impact the progenies, the future. And so we need to truly understand the positives and the negatives. See, only when you understand that there is a package and you have a lot of positives and you have also some negatives, that is when you'd start looking at it seriously. So I just saw this article and it says like China's gene edited babies may be smarter. Amazing. We all want to be smart. And then it also said that this one phrase, it's a quotation. It is actually a quotation. This was greeted with horror by the global scientific community because Dr. He and his team had actually deleted a gene in the human embryo and reintroduced the embryos. So this happened in China. So if this is going to happen, we will have to greet this with horror. This is not amazing. This is not, wow, how could you do that? Yes, of course, this is science. We're growing and we're doing great science. We're doing massive science. We're making movements and we are progressing. But what is progress? What is the defining moment in progress? Is progress changing the stuff? Just for nothing. So I also went through this article and then they talked about, oh yes, and also Dr. Chandrasegaran, he also talked about doing cosmetic stuff. See, why would you change, maybe do genome editing and why would you do stuff? Yes, to heal somebody with a serious disease which does not have an alternative. Of course you would do that. But why would you do that just because you want to have a designer? You just want to design something which should look different, which should look odd. So you should seriously think about stuff like that. So is it a boon or a bane? It completely relies in the hands of the user. And as Mani also introduced, and also like when Dr. Matheswaran, when he also spoke about it, we need to have serious policies. Not everybody should do it. Not everybody should take this into their hands. This is not a matter that can be taken in the hands of all the public, maybe just a growing or a budding artist or a scientist or a researcher. There should be strong ground rules, strong policies that should be laid, which people will have strong guidelines and they should have a license. They should be able to really understand what is going to happen when they start off something. You kick off something, you do a deletion, you do addition, you should know what is equal to what. And once you arrive at that, and yes, you know what is going to happen, fully research, prove yourself to the institution or or whatever is the body who is going to give you the license, and then you go forward. So that way we will be putting a restriction, and we can of course make gene editing a boon if we avoid the banes.
Thank you.
Thank you.
Thank you. So you raised an ethical questions on that score. Let me invite comments from Dr. Chandrasegaran. There are, particularly with respect to GMO, there were issues raised in the context of adversely affecting biodiversity, adversely affecting the agricultural ecological balance, as GMO organisms tend to produce protein secretions that could actually lead to death of the pests or insects, but then that further leads to a disruption in the ecological cycle, and therefore biodiversity can get affected is one of the complaints. The other complaint is, of course, in the genetic foods, using GMO food seeds could lead to allergic related complications subsequently, and antibiotic resistance, etc. And some of the studies are also raising issues with respect to linkage to cancer, etc. So these are GMO-related, complaints or observations or the negative points that are there. In the context of genome editing, much of this should go away, as I understand, and much of this should not apply. Biodiversity should not get adversely affected. And on the other hand, it should actually, the Green Revolution brought about what's known as the industrial agriculture, which in over 50 years now we realize it's done immense harm. Genome editing probably should get you back to what's known as not monopolistic agriculture, but multi-diverse agricultural practices, which could lead to a greater strength of biodiversity. How do you look at that?
Yeah, with regard to loss of function, that's what gene disruptions are. So that doesn't give a survival advantage to the plant. So that should not affect much, right? When do invasive organism take over? When they have some survival advantage. When you don't have that, I don't think that will be a problem. So all this gene disruption or loss of function, I don't think that will be a big problem. That's my opinion. And gain of function, you have to always be careful. Whenever you introduce, right? It's like a surgeon. The knife is in a surgeon's hand, it cures people. If it is in the wrong hand, it kills people. Right? So you just have to be cautious. You have to have your rules and regulations. If people break the rules, punish them hard. Right? And for ecology, I am not the person to comment. I'm not.
Okay.
Right.
With respect to, of course, the survivability, genome editing will offer better survivability in the context of emerging climate change impact.
Yeah, genome editing helps, right? See, in classical, even if you take dog breeding, right? When you breed a particular type of dog, it takes lots of years and years. All genome editing does, it accelerates the process, okay? And the mutations happen all the time in the world. When you walk out in sunlight, what do you think happens to all your genes exposed to UV? You get photodimers. Some of you have very good repair, you're okay. If you are, as you get older, they don't get repaired, you end up with cancer. Okay, this is part of life, evolution, right? So, I don't know how much, any process in the wrong person's hand, very bad.
Very bad, that's right.
Right? But hopefully, I think what researchers, I consider myself researchers or scientists, our approach is to do the technique as safe as possible, okay, for use in treating people. Okay. And also, if you have 1 year one, this is why I like China has taken a bold step forward regard to genome editing. They have accepted it. They are investing a lot of money into it. All you have to do is take the journals and look how many very nice papers are coming out of it. Because they have accepted it. And they are doing it. Just the simple thing. We have 1.4 billion people. We have to feed all of them. Okay? And let's do this. Right? I think India is not that. Actually, we are going to overtake it soon.
Yeah, we'll be 1.6 in another 20 years.
These things, we have to face reality, accept and move forward. Yes, but you have to strict guidelines, strict regulations, everything should be there.
What my opinion is like.
There are few questions that can be taken. If anybody has a question, please raise your hands and stand up. Yeah,
So very strict regulatory, organizations to govern, take care of this, whether it is feasible to allow for public usage or not. That should be done in this country. They should not restrict the researchers, but they should, control them. So that's what I suppose.
You cannot also overdo.
Yeah.
Right?
Okay.
that's true.
Thank you, Professor. And this is really an interesting session, like a science fiction film, what I watched actually. My question is also bit more commercial, inclined towards that. So I used to recently read some articles and also happened to watch some world news, international geopolitical news. So that I happened to see again and again there was resonating news was there. Chinese PLA is very much focused on this genetic thing, and they are going to bring some X-Men or Supermen and kind of I don't know how to call humans or creatures for their again war capacity, to increase their war capacity. Is it something real time? It is possible. Is it possible somewhere in near term? Number one. Number two question is, happen to watch the film like Universal Soldier or Species kind of films. Have you in your lab? Have you ever tried and it gave you a wrong result to an extent you have seen something which you should not supposed to see, willing to see like this? Two questions. Thank you.
Any other questions? We can take one more. Yeah, please. He'll answer that. 1 second.
So my question is regarding to Professor Chandrasegaran sir, and you told about like we can punish those if something goes wrong or if somebody overcome the limits. But what will happen like once it is released into the environment, either it is microorganism, we can't get it back. So mistake is a mistake.
I agree with that.
Yes.
Something released, then we can't.
No, can't be
But for that, you have to have all the
regulations.
We have, biocontainment 1, biocontainment 2, like COVID,
Oh, okay.
right? You have those type of research has to be done under very strict control. Okay? Yeah. But COVID also came, right?
Yeah.
Oh, yours. Please use the mic. So, I don't think, like I said, in the last 2 movies, young people should see those 2 movies. Gattaca and Boys from Brazil. Actually, Gregory Peck was an excellent actor. It's like Hitler, somebody wants to recreate Hitler, so he create a background, genetic background and its environmental background. So go watch it, it's a great movie. And the same thing with Gattaca, right? You should watch that. Yeah, so right now it's not possible, okay? You can do muscle strengthening, but it's already being done, right? All you have to do is all these weightlifters, they are taking drugs and stuff. So that's already being. To making genetic changes and doing, you cannot do it. It has to be done at the embryo level. US has prohibited it. And also, I told you challenges facing genome editing. They are real. Off target and all those things are near. It's not that easy to do. And some of the traits you are thinking about, you cannot do it by changing one gene. You have to change multiple genes. Okay? So evolution had a lot of time to do all these changes, and all organisms have come, right? Survive. But you can't just go and do them in 1 day or 2 day or 3 day. It will take a long time to do those type of things. So I wouldn't worry too much about it.
Yeah. Anyone? Any other questions?
What is your first question?
Second one was species kind of film, and it gave a wrong result. And
If you experience any results in your
There was one, when I first proposed the idea, NIH Summit grant, one of the reviewers wrote, we get our reviews back, what they tell you. So one guy said, this is a nice idea, it won't work. But he's a young investigator, we should fund it. So they funded it. Then I wrote another proposal. A few years later, after that expired, it went back to the same guy. Now I had proposed something new. He wrote back. Last time I reviewed, I said it won't work. He proved me wrong. Now he is proposing something new. I'm not going to say it won't work. So give him the money. So that's US. That's the difference.
Fantastic. That's right. Yeah. Anyone? Any more? Okay. Just to answer, please go ahead, Mr. Gopal.
Professor, after the success of BT cotton, it caught the attention of the people. Then immediately came BT brinjal. There was big hue and cry. Government was against it. But it has not been stopped. It has been silently happening. There are a lot of genetically modified foods. Silently it is being produced and it is coming to the market.
Yeah.
So that should be a proper management. This for adulteration there is a check. For quality there is a check. Whether these are all the things denied, whether it is flowing into the market, to check there is no agency at all. What do you say?
I'll just add on to that. Much like in any other field, now the American system works on patents and capitalist model. So Monsanto was dominating or monopolizing the BT cotton, and they would have done it in the others as well. But Indians being what they are, they quickly found alternate routes to take the same gene solutions, and Monsanto went to court, but they couldn't do anything. But it's dangerous because the illegal gene editing processes were coming in, and the government is aware of it. They're bringing in some kind of legislation on that.
I agree. There is always people who will try to come through, right? When there is money, people do strange things. That's true here too. If you can bring it and do things. But it also comes through Bangladesh, if I'm right? They have no restrictions, I think. So they allow, so then the border is porous, you can get it. So, but you can get rid of the print. Genome editing can remove all of those. It's not that hard to do it.
Yeah, please go ahead.
Well,
Last question. Yeah.
I am Ramesh from MMA. Well, nice to see some tomatoes coming up from Pondicherry, and also heard from Mr. Mani that some peanuts are coming up with bigger sizes, good for my pocket as well. Like that, Mr. Chandrasegaran, have you come across anything substantive, which is good, which is wonderful, which is happy to hear in your own experience as a researcher, at least 3 things which has actually impacted our lives in a very, very, very positive way, and not like what Atesh has told in the form of coming out with some demons kind of a material from China kind of a thing. That's negative. Positive things, 3 things can you say by which we can go home happily, along with Judy, Gopal.
Okay. So the talent, right? They have been used to cure cancer in 2 girls at least. Okay? There is a newspaper article on that. Yes. So it was used to treat a blood cancer. Okay, so it's there, but they have tried more people, it works in some people and not others. Okay, that's one thing. Second of all, I think your food, already you can buy all those mushrooms, which is already in US. Like you said, a lot of them have come. Third thing, they're trying to do genome editing without taking cells out within the body. And some people suffer from the blood defect in factor 7, factor 8, which clots blood. If you don't have it, you'll continue to bleed. Now they have done it in vivo, it seems to be helping. So these things are all in preclinical trials. They will be coming out. But definitely those 2 girls are fine. And recently there was another girl who was treated for sickle cell using CRISPR. She's doing fine after 2 years. Okay, no problems associated with sickle cell. Yes, but like anything, in the beginning they are very expensive because they want to get back all the money. And also these, unlike US, I want to make it clear, US is a capitalism country. They want money, they are willing to work very hard, okay? But they are expected in return, right? They want, so in the beginning, so they have a pattern, they want to make as much money as possible within that pattern. After that, it becomes very cheap, right? But it's not their fault also, it's also whoever agrees to this WTO. If your government doesn't agree, there is nothing they can do, right? But you also agree and sign the agreement. So, yeah, so you have to negotiate with them, try to get them cheaper, or the other way is you may have to negotiate for the whole country, right? See, when you negotiate with a large population, then you can get it cheaper. That's what I'm trying to say.
That's wonderful. And just to close on a final point, science is always progressive. It does, at the end of the day, it does good for humanity. But the question that you asked is also important because you're talking about using these technology as a bioweapon. And I would like to quote Plato, who said 2,500 years ago, only the dead have seen the end of war. Okay? So Hitler, when he tried to control his population to the defining the best Germans, and euthanasia he was actually implementing, he was trying a form of control, right? So it's only a matter of time, as he pointed out in the challenges, that this will also become a weapon in the hands of few countries and few people, and we got to guard against that. So we've run out of time. It's an excellent session, and it's for me to thank Dr. Chandrasegaran, Dr. Judy, Gopal, and Dr. Manikandan. Please give them a big hand. Thank you very much.
Thank you, Marshal. Again, a fascinating thing. We did this because we want to do some genome editing for each one of you to learn something. Keep attending MMA event. A lot of editing due to your brain and a lot of knowledge. Thanks so much, and I request Atish to join us, the president of Memento. I think viewers, more than 400 people are watching. Thanks for joining us this evening. Come.